everyone and welcome to Miss Estruch Biology. In this video we're going through topic 15, control and coordination. This is everything you need to know for this topic, and if you do want that all written in one place for you, then check out my A-level notes which cover all the theory for CIE, key terms, key marking points, and you've got examiner's tips included there as well. So those are linked below for you to help boost your A-level grade. But for now, let's get into the content. Topic 15 then, and it's all to do with control and coordination. So in chapter 14, we saw how homeostasis occurred to help regulate blood glucose levels and water potential, and you can actually have a look at that previous video as well. But in this video, it's going through how you can also have coordination and control using the nervous system. And with the nervous system, it's going to be far more rapid rather than gradual, which is what the hormonal response is. So insulin, glucagon, ADHD and adrenaline are examples of hormones which act as signaling molecules released into the bloodstream, and they travel long distance from where they're produced to their target organs. And these hormones are produced within endocrine glands, which basically means a gland that produces a hormone, and groups of cells responsible for secreting one or more substance directly to the bloodstream. Endocrine glands lack ducts, and the endocrine system encompasses all those ductless glands within the body, including the pituitary gland, which we can see at the top just here within the brain. So if we have a look at the nervous system compared to the endocrine system, I've put together this summary table for you so you can screenshot it, print it out and put it into your notes, or maybe turn it into flashcards. But you need to know the comparisons between the mode of communication, so how that information is delivered, electrical impulse versus hormones, the speed, duration of the effects, the target tissues, response time, adaptation to stimuli, regulation of functions, organs involved, mechanisms of actions, and feedback regulation. So these are all just key facts to know. So I'm not going to talk through all of this. You can screenshot and keep that in your notes. So moving on then, just focusing on the nervous system. The nervous system is made up of billions of neurons. These can be categorized into three main groups: sensory neurons, relay neurons, and motor neurons. And these three specialized cells do still have some features in common. So they all have a cell body, which contains the organelles that you find in a typical animal cell. So for example, the nucleus and the mitochondria. They have dendrons, which carry the action potentials to surrounding cells, and they have an axon, which is a conductive long fibre that carries the nervous impulse along the neuron. So if we then have a look at the actual structure we can see, so we talked about some of those. Here's our cell body. We've got the nucleus inside of that cell body. This part is the axon, and we can see the dendrons branching out here as well. Myelinated neurons have Schwann cells as well, which is shown here in yellow, wrapping around the axon. And they wrap around to form what's known as the myelin sheath. And this is a lipid, and therefore it doesn't allow charged ions to pass through it. And in that way, it insulates and prevents the electrical impulse from travelling the entire length of the axon. Instead, has to jump from node to node, which is known as the node of Ranvier, and that speeds up the conductance. So that's what we've got here, that those gaps are called the nodes of Ranvier, and we get that action potential jumping from node to node, which is known as saltatory conduction, and that is what speeds up how quickly that impulse can travel. So if we have a look at a sensory neuron, these are the neurons responsible for carrying electrical impulses from the sensory receptor cell to the relay neuron, sometimes also to the motor neuron and the brain. They have a long dendron which carries the impulse from the sensory receptor to the cell body, which is what we can see here, and there is the cell body, and then an axon to carry the impulse from the cell body to the next neuron. The intermediate neuron, sometimes known as the relay neuron, these neurons carry impulses between the sensory and motor neurons, and they have multiple short axons, which we can see here, and also dendrons branching off each of those. And then finally, the motor neuron, these carry impulses from the relay or sensory neurons to the effector, and the effector is either a muscle or gland, that is the part that carries out the response. And these have one long axon and multiple short dendrons. A sensory receptor then, these are cells that detect a stimulus. That is what a receptor is. And these cells are transducers, meaning they convert different types of stimuli into electrical impulses. There are different types of sensory receptors, and each type can detect a different stimulus. And here is your table summarizing the different types of sensory receptors and the stimulus that they detect. So photoreceptors, photo meaning light, they detect light, and that would be your rod and cone cells in your retina of the eye. Thermoreceptors, thermo means temperature, so they can detect changes in temperature or heat energy. Mechanoreceptors, so the Pacinian corpuscle and the sinus corpuscle as well, detect the pressure. Taste, smell and balance as well. So taste and smell are chemical, balance is movement. Next time we move on to the actual changes that happen in a response. And when there isn't a stimulus, your neurons would be in resting potential state. So that's what we're going to go through first. When a neuron is not conducting an impulse, meaning there's no stimulus, there is still a difference between the electrical charge inside and outside of the neuron, and that is known as the resting potential. You actually have more positive ions, which are the sodium ions and potassium ions, outside of the neuron compared to inside. And therefore, the inside of the neuron is comparatively negative at minus 70 millivolts. So this graph here is showing you an action potential, which is what happens when there is a stimulus. But this section here of the graph, this is before there is any stimulus. And we can see that the voltage, which is our potential difference, is remaining at minus 70 millivolts. So the way that that occurs then is there is a sodium ion, potassium ion pump, and this stage requires ATP for active transport. And this co-transporter pump will pump 2 potassium ions into the axon of your neuron and 3 potassium ions outside of the cell. That creates an electrical chemical gradient because we'll then have more potassium ions inside of the cell and more sodium ions outside of the cell. And as a result of those electrochemical gradients, potassium ions will diffuse out of the cell through these channel proteins, and sodium ions will diffuse into the cell through their channel proteins. However, there are more potassium ion channels than there are sodium ion channels, so we say that the membrane is more permeable to potassium ions. Also, the potassium ions are permanently open and some of the sodium ion channels are not permanently open. So as a result, you get more potassium ions diffusing out than you do sodium ions diffusing in. And as a result, you have more positive ions outside compared to inside. And that is how we get this minus 70 millivolts, or the resting potential. So if there is a stimulus, that can result in an action potential. And this is when the neuron's voltage, or potential difference, increases beyond a set point from that resting potential. So our resting potential is minus 70 millivolts. But if we actually get a change that brings the voltage up to the threshold point of minus 55 millivolts, then you will get an action potential occurring. So an increase in voltage, also known as depolarization, is due to the neuron membrane becoming more permeable to sodium ions, meaning more sodium ions are going to diffuse into the axon. And because those have a positive charge, that will bring that voltage to become more and more positive. So we go from minus 70 up to minus 55, which is more positive. Once an action potential is generated, it moves along the axon like a Mexican wave. So you'd have to generate that action potential at this point on the axon. That would then trigger an action potential at this node of Ranvier, and then this one, this one, this one, this one. And that's what we mean by the movement being like a Mexican wave. So let's go through then how an action potential is generated. We've got our resting potential here, where we have got those voltage-gated sodium ion channels closed. What that means is these are sodium ion channels that sodium ions can diffuse from the outside to the inside of the cell. However, those channels only open when there is a particular voltage, whereas we have some potassium ion channels which are permanently opened. So that's what's happening at our minus 70 millivolts for the resting potential. But if there is a stimulus, that stimulus causes some of the voltage-gated sodium ion channels to open. And as a result, sodium ions are going to diffuse in. And that causes the voltage to become more positive. Now, if you have a large enough stimulus that will cause enough sodium ion channels to open to reach the minus 55 millivolt threshold. If it's only a small stimulus, that won't cause enough of those voltage-gated sodium ion channels to open, so you won't actually get an increase in the voltage to the threshold of minus 55 millivolts, and therefore you wouldn't respond to the stimulus. And that's actually really useful because you don't want to respond to every slight change because that would overwhelm your senses. So you're only going to respond if the pressure or the sound or the smell is strong enough that it does deem worthy of responding to, so that it would actually protect you from potential harm. Now, when you do get to that threshold point of minus 55 millivolts, that causes even more voltage-gated sodium ion channels to open, and therefore you get even more sodium ions diffusing in, making that voltage now become positive, and it peaks at plus 40 millivolts. Now, when you get to that point, that then causes those voltage-gated sodium ion channels to close, but we still have potassium ion channels that are permanently open. So we now have positive ions being diffusing out, so they're going to leave the axon. So we then start to become more negative again, which is known as repolarization. And at this point, we have even more potassium ion channels open, so even more potassium ions are diffusing out, which actually results in this overshoot where the voltage becomes even more negative than the resting potential. And we call this stage hyperpolarization, where the voltage can go to minus 80 millivolts. And this is known as the refractory period as well. During this point, these voltage-gated sodium ion channels are temporarily unable to open. So you get a very short period of time where a new action potential wouldn't be able to be generated. And we're going to talk about why that is an advantage. But for now, that is just showing you how that depolarization results in this action potential, and then how that afterwards results in repolarization going into that refractory period. But then, because of that sodium ion, potassium ion pump that we talked about in maintaining the resting potential, it does bring that voltage back up to the minus 70 millivolts of resting. So this is just what I meant by the Mexican wave. That action potential is generated at this point on the axon. That then jumps to the next node of Ranvier, the next one, and that will happen all the way along the axon until it reaches the end of that motor neuron, and then we get to a synapse. So the all or nothing principle is this concept that if the depolarization does not exceed that threshold of minus 55 millivolts, then you will not get an action potential. So nothing happens. That's the nothing part of all or nothing. Any stimulus that does trigger depolarization, meaning it has reached that minus 55 millivolt threshold, the maximum voltage is always the same at plus 40 millivolts. So a bigger stimulus doesn't result in a greater peak, a greater voltage. It's always going to be a maximum of plus 40 millivolts because of the set number of channel proteins present and the number of ions present. But what does vary is the frequency at which those action potentials are going to be generated. So the larger the stimulus, the quicker the frequency, or the higher the frequency. So this is what we mean by the all or nothing principle. If you don't reach that minus 55 millivolts, then nothing will happen. You won't get an action potential. But if you do, then you'll always get an action potential, and it will always peak at that same voltage. So it's important, as this makes sure that animals only respond to a large enough stimuli. And this is what I was talking about when we mentioned the threshold potential earlier. You have to have a large enough stimulus to reach that threshold, and therefore you're only going to respond to large enough stimuli and not every slight change in your environment, because that would overwhelm your senses. So a bit more about the refractory period. After an action potential has been generated, the membrane enters that refractory period when it can't be stimulated again. And that's because those sodium ion channels are recovering and they can't be opened. So that is important because it ensures that discrete impulses are produced. An action potential cannot be generated immediately after another, and that is why it makes sure that each impulse is separate. It also ensures that action potentials only travel forwards in one direction. It stops the action potential from spreading out and going backwards. And if it did go backwards along the axon, that would prevent it causing a response because it wouldn't actually end up reaching the effector. And lastly, it limits the number of impulses, and that's important to prevent an overreaction to a stimulus. So how you can then speed up these impulses? One way is the Schwann cells, which we briefly talked about. Schwann cells wrap around the axon to form the myelin sheath, which does not allow the charged ions, those sodium ions and potassium ions, to pass through, going in and out of the axon. There are those gaps, which are known as the node of Ranvier, though, and that results in the saltatory conduction, which is where that action potential jumps from node to node. And that means the action potential travels faster along the axon because you're not generating an action potential at every single position along the axon. So when that impulse does reach the end of a neuron, it reaches a gap, which is known as a synapse. So the synapses are these gaps between the end of an axon of one neuron and the dendrite of another one. And at that point, the action potential is transmitted as neurotransmitters, which are chemicals, which diffuse across the synapse. So let's go through how that happens. When that action potential reaches the end of the neuron, that is known as the synaptic knob, the end of the neuron. And depolarization of the synaptic knob causes calcium ion channels embedded within the membrane to open, and therefore calcium ions diffuse into the synaptic knob. Those calcium ions cause vesicles within the synaptic knob, which contain the chemicals, the neurotransmitter, to move towards the cell surface membrane. They fuse with that presynaptic membrane, and when they fuse, that causes the release of the neurotransmitter by exocytosis into the synaptic cleft, which is what we call this gap between the two neurons. Because that neurotransmitter is only being released from the presynaptic neuron, we have a concentration gradient. So that means the neurotransmitter chemicals can diffuse down their concentration gradient, which causes them to diffuse across the synaptic cleft to the postsynaptic neuron. And it goes to the membrane of that postsynaptic neuron, which has receptors embedded within that neuron membrane. And those receptors are complementary in shape to these neurotransmitter chemicals. And as a result, the neurotransmitters bind to those receptors. Once they bind to those receptors on the postsynaptic membrane, that causes sodium ion channels on that postsynaptic membrane to open. And once those sodium ion channels open, sodium ions can diffuse into the postsynaptic neuron, causing the postsynaptic neuron to depolarize. And that depolarization can then result in the generation of an action potential, and that has then triggered an action potential in the next neuron. Those neurotransmitters do not remain permanently bound to the receptors though, because if they did, that would result in the action potential constantly being generated in the postsynaptic neuron, even if there was no longer a stimulus there. So that means the neurotransmitter has to be released from the receptor. And the way that that happens is the neurotransmitter chemicals are broken down or degraded, and that causes them to be released from the receptor. That causes the sodium ion channels to close, and that is how you then don't get any more action potentials being generated. And that broken down or degraded neurotransmitter can be recycled and returned back to the presynaptic neuron to then regenerate those neurotransmitters to be reused again. So this response is unidirectional. It's unidirectional for a few reasons. First of all, you only have the vesicle containing the neurotransmitter in the presynaptic neuron. You only have the receptors that the neurotransmitter binds to on the postsynaptic neuron, and the neurotransmitter diffuses across because of the concentration gradient. Now, an exact example of this synapse response or transmission across the synapse is a cholinergic synapse. And it's called that because the neurotransmitter is acetylcholine. So you would go through that exact same process that we described, but wherever we said neurotransmitter, you'd say acetylcholine instead. And the acetylcholine is broken down or degraded after it's attached to the receptors by the enzyme acetylcholine esterase. And that breaks down the acetylcholine into choline and acetate, which then gets recycled. Now, thinking back to when we talked about the generation of an action potential, we said that you only get an action potential if depolarization reaches at least minus 55 millivolts, that threshold potential. So when you're at a synapse, summation has to occur to reach that threshold. And summation is the rapid buildup of the neurotransmitter in the synapse so that you have enough neurotransmitter binding to enough receptors to open enough sodium ion channels to result in sodium ions diffusing in to reach that minus 55 millivolt threshold. And there's two ways that summation can occur: spatial and temporal. Spatial summation is when you have multiple presynaptic neurons all converging at one postsynaptic neuron. And collectively, those presynaptic neurons should release enough neurotransmitter to help trigger an action potential. Temporal summation is when there is only one neuron, so one presynaptic neuron converging to one postsynaptic neuron, but that one presynaptic neuron will release neurotransmitter repeatedly over a short period of time to add up to enough to exceed that threshold value. Now, at the end of your response, you have an effector, which can be a muscle cell or a gland, and that means you'd have your motor neuron reaching a muscle fibre. And the gap between the motor neuron and the muscle fibre is called a neuromuscular junction, because you've got a neuron reaching muscle. So these are specialised synapses, which we said are formed between the motor neuron and the muscular fibre. And when an action potential reaches the end of the motor neuron, it triggers the release of neurotransmitter acetylcholine into the synaptic cleft, just how we saw that happens in a typical synapse. That acetylcholine then binds to receptors, but this time the receptors are on the muscle fibre, not on a postsynaptic neuron's membrane. And that then causes sodium ion channels to open, sodium ions diffuse in, and it causes depolarization of the muscle cell membrane. This depolarization generates an action potential that spreads across the muscle fibre membrane and into tubes known as T-tubules, and it gets into the T-tubule system. So you could be asked to compare similarities between a neuromuscular junction and a cholinergic synapse. So here is our comparison. They both involve unidirectional movement. A neuromuscular junction is always excitatory, meaning it will always result in a response, whereas cholinergic could result in a response, or you do get inhibitory synapses where chloride ions are released instead. We've got the difference between where they connect. The neuromuscular junction is at the end of that response arc, whereas a cholinergic synapse can result in a new action potential generated in the next neuron. And then lastly, the acetylcholine binds to receptors on muscle fibre membranes for a neuromuscular junction, whereas the acetylcholine binds to receptors on postsynaptic membranes for a neuron. So at that neuromuscular junction, we said that the action potential causes depolarization that reaches the T-tubule system. So T-tubules are these invaginations of the sarcomere, which is the muscle cell membrane, that penetrate deep into the muscle fibre. And they ensure the rapid transmission of the electrical signal throughout the muscle fibre, enabling uniform initiation of muscle contraction. As the action potential travels along the sarcomere, which we can see up here, and into these T-tubules, which are shown there, it simultaneously activates the entire muscle fibre. The sarcoplasmic reticulum, or SR, is a specialised form of endoplasmic reticulum found in the muscle cells, and it is crucial in regulating calcium ion channels. And those sarcoplasmic reticulum surrounds the myofibrils, which are the contractile units, which we're going to have a look at, which store calcium ions. So when the action potential travels along the T tubules, which are here, it triggers the release of calcium ions from the sarcoplasmic reticulum into the cytoplasm of the muscle fibers, and this increases the calcium ion concentration, which leads to the exposure of binding sites on the actin filaments, which ultimately results in a muscle contraction. So that leads us on to what muscles, or the structure of muscles, and how we get that contraction. So muscles always act in antagonistic pairs, which means there's one contracts, the other relaxes, and that happens against the skeleton to result in movement of your bones. Myofibrils are made up of fused cells that share nuclei and cytoplasm, which is known as sarcoplasm. And there is also a really high number of mitochondria because for a muscle to contract, you need ATP. So here is a myofibril, and the myofibrils are made up of sections known as a sarcomere. Muscle fibers are made up of millions of myofibrils, which collectively bring about the force to cause the muscle to contract. And myofibrils are made up of two key proteins. We have myosin and actin, and those form the sarcomere. And that's what we're going to have a look at in more detail. So this is just showing you that sarcomere. If I go back to here where we can see this whole, or a section of a myofibril, and we've got our sarcomere indicated at this point with a thin filament and a thick filament. The thick filament is the myosin, which is one of the proteins. The thin filament is the actin, which is the other protein. And this time it's shown here in blue. That is your myosin, the thicker filament. And in red here, we have the actin, which is the thin filament. And it's just named that because they are thicker and thinner proteins. So we can see here in the sarcomere, we have the actin on both sides, and that is interleaved between the myosin, which is in this fixed position. So the myosin stays in that fixed position, but when a muscle contracts, the two sides here with the actin slide closer together and they slide over the myosin. And we can describe this contraction based on these different lines and zones in the sarcomere. So the A band, that is the section of the sarcomere where you have the myosin. And because the myosin doesn't change position, the A band will always stay exactly the same width. The Z lines, which we can see here, those indicate the parameters or the border of your sarcomere. And when a muscle contracts, the Z lines get closer together. So we can see that Z line's moved in here and it's moved in there. So those Z lines get closer together. That's why when muscles contract, they actually get shorter and fatter because the Z lines are moving closer together. We then have this I band. The I band is where you just have actin by itself, not overlapped with any myosin. And when a muscle contracts, that I band gets shorter because the actin is sliding over the myosin, so you have more section where it's an overlap. The H zone is where you just have myosin by itself with no actin in between it. And when the muscles contract, because the actin is sliding in between, you have a shorter H zone. And those are your key sections that you need to know. So you need to be able to label those bands and zones, and you need to know what happens to them during muscle contraction. And we're going to go through how the actin slides closer together and over that myosin in this muscle contraction. And this is what the sliding filament theory is. When that action potential reaches the muscle, we already talked about how it generates that depolarization traveling through those T tubules, and it stimulates a response because it causes calcium ions to enter, and those calcium ions can cause tropomyosin, which is shown here in orange as protein, to move to a different position. And that position that it moves to results in the actin exposing the myosin binding sites. So that thin filament actin has binding sites on it that myosin heads, which are these structures branching off the myosin, they can bind to. So once those calcium ions have been released and the tropomyosin has been pulled out the way, the myosin binding sites are revealed, and the myosin heads, because they're complementary in shape, those attach. And whilst the ADP and PI are attached to that myosin head, they are the correct shape, that is the myosin head, to be able to attach to those myosin binding sites on actin, and it forms what we call a cross bridge. So we've now created a bridge between the myosin and the actin. This angle that is created in the cross bridge creates tension on the myosin head. And as a result, that tension results in this power stroke, which is when the myosin head flicks. It really slides that actin along the myosin. And in doing so, that power stroke action causes the release of ADP and PI. An ATP molecule can then bind to the myosin head, and at that point it causes the myosin head to slightly change shape so that it's now no longer complementary to the myosin binding sites, and as a result it detaches from the myosin binding sites on actin. Within the sarcoplasm there is an enzyme ATPase, which is then activated by the calcium ions to hydrolyze that ATP on the myosin head into ADP and PI, and that releases enough energy to cause the myosin head to return back to its original position, which is known as recocking. So cocking of the myosin head is the movement of it back to its original position. And as long as the calcium ions are still present, holding that tropomyosin out the way, that whole process can continuously repeat, and that will slide the actin closer and closer together until you have a full muscle contraction. So the final part of this topic is looking at the control and coordination in plants. And plants can't run away from animals. They don't have muscles, so they can't move to the shade or move to the light like animals have evolved to be able to do this. So instead, they have a different range of responses to defend themselves from herbivores trying to eat them and abiotic stresses. So the Venus flytrap is an example, and this shows a rapid response to stimulation, particularly when prey, such as insects, come into contact with its modified leaves, known as lobes. And these lobes are lined with tiny sensitive trigger hairs, which you actually can't see in this diagram, but on the red section here, that is where you'd have those trigger hairs. And when an insect brushes against these trigger hairs, it causes the hairs to bend, generating an action potential within the plant. The action potential is similar to the nervous impulses found in animals, and they travel rapidly throughout the lobes. The action potential spreads through specialized cells within the lobes called motor cells, and these cells act as a bridge between the trigger hairs and the closure mechanism of the trap. And as the electrical signal reaches the motor cells, they rapidly transport ions, such as calcium ions, across the membranes, creating a change in turgor pressure of the cells. And that change in turgor pressure within the motor cells causes a rapid change in the curvature of these lobes. And that is why the lobes end up snapping shut, and as a result, it traps the prey inside. Once trapped in that closed section, the plant secretes digestive enzymes to break down the captured prey, and that extracts nutrients from the insects, such as nitrogen and phosphorus, from their bodies. And that provides the plants with essential nutrients, which the environment may not contain because there might be nutrient-poor soil. So other responses are tropisms, and this is the term given to when plants respond via growth to a stimulus. You can have positive or negative tropisms. Positive means it's growing towards a stimulus. Negative means it's growing away from the stimulus. And plants typically respond to these three stimuli: light, gravity, and water. Tropisms are controlled by specific growth factors, and one key example is IAA. And that's a type of auxin that can control cell elongation in the shoots of the plants, and actually increases cell elongation in the shoot, and it inhibits the cell elongation in the roots. So if we have a look then at phototropisms, which is an example where it's responding to light. In the shoot, light is needed for the light-dependent reactions in photosynthesis and the leaves, which are going to be branching off the shoots. So it's an advantage, an adaptation, that the plants, the shoot part of the plant, will bend towards the light source, a positive phototropism. And the way this happens is IAA is produced in the shoot tip. The IAA is able to diffuse to other cells where it isn't produced and cause cell elongation. And if there's unilateral light, the IAA will diffuse towards the shaded side, because unilateral means the light source is only on one side, which is what we can see here in image 2. The light source is slightly over to the right. So all of that IAA is going to diffuse to the shaded side, which is on the left. That will mean we'll get more cell elongation on the left-hand side compared to the right. So the left side of the plant is going to be longer than the right-hand side. And as a result, the plant starts to bend towards that light source, which is useful because the plant will then get more light energy, so it can absorb more of that light energy for the light-dependent reactions in photosynthesis. Now, in the roots, we have the opposite effect. Roots do not photosynthesize, so they don't require light, but instead it's an advantage if the roots are deeper in the soil so they can anchor in, and also it helps to reach more water. So in roots, if there's a high concentration of IAA, it inhibits cell elongation, and that causes the root cells to elongate more on the lighter side, and so the root ends up bending away from the light source, which is what we can see here in this animation, and that is negative phototropisms. Gravitropism, this will then occur in the roots and shoots differently. In the shoots, we can see that the IAA diffuses from the upper side to the lower side, so more of it's now on the lower side, so it goes down with gravity. That means that we get more cell elongation on the lower side, and that causes that side to be longer, and therefore it bends upwards. And in this case, it's a negative gravitropism because the plant will be growing against gravity. In the roots, IAA moves to the lower side of the root, so that the upper side elongates, and the root bends down towards gravity and anchors the plant in. And this is a positive gravitropism because the root is bending towards the stimulus, which is gravity. So plant hormones then control a range of responses in plants, such as the ripening of fruit, germination of seeds, and as we just saw with the IAA, the lengthening of stems and when also leaves drop. Examples of plant hormones include auxins, which is what we just went through with IAA, ethene, which is a gas that causes fruit to ripen, and gibberellin, which stimulates seed germination, stem elongation, and pollen tube growth in fertilization. So a bit on gibberellins then. Gibberellin is needed for germination of barley seeds, as an example, by initiating and coordinating the physiological processes for growth. When a barley seed absorbs water, it triggers the synthesis and release of that plant hormone gibberellin. And these gibberellins act as signaling molecules within the seed, resulting in a series of events that leads to germination. One of the primary functions of gibberellin during germination is to stimulate the production and activation of enzymes involved in breaking down stored food reserves in the seed. And barley seeds store energy in the form of starches and proteins. Gibberellins prompt the release of enzymes such as amylases and proteases, which will then hydrolyze those stored reserves into smaller, more accessible molecules. And this breakdown by enzymes provides the embryo plant with necessary nutrients and energy sources, such as glucose and amino acids, to fuel the metabolism during germination. Additionally, gibberellins promote cell elongation and expansion, which are essential processes for the emergence of that embryonic shoot, which we can see in the image here. And by stimulating cell elongation, gibberellins enable the embryo to push through the seed coat and emerge to the surrounding soil. And this growth is crucial for establishing young plants and enabling it to access light and nutrients for further development through photosynthesis. Gibberellins also play a role in overcoming seed dormancy, which is a state of suspended growth and metabolic activity that prevents germination if there are unfavourable environmental conditions. So gibberellins counteract the inhibitory effects of other hormones such as ABA, which maintains seed dormancy. And by promoting germination, gibberellins help the seed to respond appropriately to favourable environmental cues such as moisture and temperature, and therefore initiate the growth process. Auxins play a key role in cell elongation, like we saw, and this is by stimulating proton pumping to acidify cell walls. And this process, known as acid growth, is essential for cell elongation and expansion in various plant tissues such as the stems, roots and young leaves. So the auxin promotes the activity of proton pumps, particularly H+ in the ATPases, and that is located in the plasma membrane of plant cells. These proton pumps actively transport hydrogen ions, which are protons, from the cytoplasm into the cell wall space or apoplast. And as protons are pumped into the cell wall space, they accumulate, leading to a decrease in pH, which is the acidification of the cell wall. This acidification softens the cell wall by disrupting the bonds between cellulose microfibrils, hemicelluloses and pectins, which are the main components of the cell wall matrix. That acidification of the cell wall increases its plasticity, making it more flexible and susceptible to deformation. This softening allows the cell to expand more easily under turgor pressure, generating the influx of water into the cells, and as a result, the cell elongates. And that's how we get that cell elongation. So that takes us to the end of this topic. Hope you found it helpful. If you did, don't forget to subscribe so you don't miss out on any of the latest videos.